So we've covered mitosis in quite a bit of detail, talking about the different phases of mitosis. This diagram I really like because it's a good way to recap sort of the process of mitosis and also use it to bridge into the concept of meiosis. We're going to go through meiosis, but not with as much detail on all the different steps. We're going to focus on how it's the same and how it's different, and also how it's related to producing the gametes that are used for sexual reproduction. And so down here would be gametes, because again, that's the whole name of the game for meiosis. That's the only purpose of meiosis in humans. And so mitosis, just as a simple review here, we had interphase is not illustrated, so interphase. And the interphase of both mitosis and meiosis are the same because we've got this chromosomal duplication happening in both. Both of these are for cell division, but they have different functions. So mitosis was for growth and repair. I guess reproduction if you are a single-celled organism, but humans aren't, where meiosis is for this production of gametes. And so they have different purposes. Prophase, metaphase, prometaphase is not illustrated here, anaphase, telophase, and then this would be the cytokinesis down here. And so what we have going on is we could talk about this being the original cell. And so we talk about it as being a diploid cell. It's diploid because it's doubled, right? It has a full set of chromosomes. In humans, the diploid cells have 46 chromosomes. Now that's different in different organisms or different animals and different plants, but for humans, this idea that diploid cell has a full set of chromosomes. So we have this duplication going on, and then we could follow through here back down to diploid cells. These daughter cells should be the same as parents. So directing our attention to the other side, this meiosis, how things are different over here, it's going to go through meiosis 1 and meiosis 2. It's going to go through 2 rounds of those phases of division like metaphase did. So they're similarly named, but we do have prophase 1 and prophase 2 and metaphase 1 and metaphase 2 and on the way because we've got meiosis 1 and meiosis 2 here. Now, meiosis 2 is a whole lot, like this process down here is a whole lot like this process over on this side. Because if we take a look at these cells, they are akin to our original. And so this process down here is a lot the same. This is different. So meiosis 1 has some very different things happen. It's different because how the chromosomes line up. So when we take a look at metaphase and metaphase 1, right, mitosis and meiosis, meiosis 1 specifically, we have the chromosomes lining up singly. So they line up singly. I don't even know if that's a word. And single file if you want. In metaphase 1, in meiosis, we have them lining up in pairs. And so these pairs we could call tetrads. They could also be called homologous chromosomes. And so tetrads are homologous chromosomes or they're pairs of chromosomes. And this process right here goes from a diploid cell, a full set of chromosomes, to a haploid cell. So in humans, these would be sperm and egg, and they are haploid. And so haploid in humans is 23 because when we have a sperm and an egg come together, we want 23 and 23 to make a full human again. So lots of scribbling on that slide. Don't worry, it's all going to come back to you again. I put it all in the coming slides, but I really like this slide because it's one stop with a lot of information in it. And it also has images in it that help sort of put these ideas into pictures, which I like for my brain. So some facts about meiosis before we get started here. So kind of like we had all of those words that started with the letter C. Meiosis is different in that we have somatic cells and we have gametes. And so these kinds of cells are different. So somatic cells are your body cells, and those would be produced like your ptosis through mitosis. And in any somatic cell, you would have pairs of chromosomes. So you have one chromosome that came from your mom and one chromosome that came from your dad. And these pair up into homologous chromosomes, homologous pairs, if you will. And so humans have 46 chromosomes. That's their diploid number. That's their full set. And 23 from the egg and 23 from the sperm. And these were haploid. The fancy name for these are gametes. So gametes are through meiosis, and somatic cells are through mitosis. So the chromosomes are matched in homologous pairs or a tetrad. So those words are somewhat interchangeable. And they're matched in how long they are, where their centromere position is. So is it up high? Is it a high-waisted one? Or is it, and I would draw the homologous chromosome would match it, right? Or is it a really balanced middle centromere, right? So they're matched in size, where their waist is. How do you spell waist? Where their middle is. And then also for what they're coding for. And so if you have the gene here for eye color, well, the homologous chromosome is also going to be coded for eye color right there. And so these gene locations are loci, or this would be one locus, right? Singular. So the loci is the position of the genes. You have different versions found at the same locus on maternal and paternal. So maybe your mom has blue eyes or brown eyes, let's say, and your dad has blue eyes, right? And so this is what's coding for eye color at that locus or loci. If you were to take any somatic cell in your body, so any skin cell, that cell that you looked at in your cheek cells in the lab, and were to separate out all of the chromosomes and line them up in their homologous pairs, and then take a picture of them, so you line them up from tallest to smallest, and you number them. So the biggest one gets to be 1, the next one gets to be 2, on down the line here. They're matched in their centromere location and their loci, what they're coding for. And you can see that's what's trying to be illustrated on this with the coloring. And so these homologous pairs are lined up from biggest to smallest, and we'd get 22 autosomal chromosomes, and those code for everything about you other than your biological sex. And the 23rd pair here, that this would be number 23, are your sex chromosomes. And so males are typically XY and females are typically XX. And so if you're a biologically female, you have 2 X's in that 23rd location. If you're male, you have an X and this little snippet of a chromosome here, which is a Y chromosome. It's not as fully formed basically as an X, and it's so small it doesn't carry as much genetic material as we're going to talk about in sex-linked traits. And so this is a karyotype. It's an image of stained chromosomes arranged in homologous pairs from biggest to smallest. And so it really is a snapshot of all your genetic information. The homologous pairs that are lined up, this one might have come from your mom, and this one came from your dad. And they look like straight lines here, but you can imagine these are just kind of like squished together. So I usually stand like with my arms and legs out, like kind of like a starfish, but these ones are more like they're kind of, your arms and legs are close together here. So this is an X and this is an X and this is an X, as this is an X and this is an X. I should have switched colors. We've got this idea that we have autosomes and sex chromosomes in our somatic cells. So contrary to some autosomes or somatic cells would be gametes. Sorry, contrary to somatic cells, are gametes. And gametes are the sperm and the egg in humans. So they have only a single set of chromosomes. They're produced through meiosis, and it creates haploid cells. And so diploid cells, I've gone through this, but it's now written out for us. So diploids have a full set of chromosomes. In humans, 46. Haploid cells, the sperm and the egg in humans, have 23. And so we have mitosis, creating somatic cells. And in those somatic cells, you have autosomal chromosomes and sex chromosomes. And then in haploid cells, you still have autosomes and a sex chromosome, but you only have half. You don't have pairs here. You don't have homologous chromosomes. And so meiosis is happening in the sex organs, in the testes and the ovaries, and it makes the gametes, the sperm and the egg. This is different. So meiosis makes the stuff we need for sexual reproduction. Sexual reproduction is actually where the union of the sperm and the egg, where they come together. So fertilization is that union, and then that's going to create the zygote, which will have a lot of mitosis happen to create a multicellular baby. So this is the life cycle in humans, and so you have mitosis, from this idea of conception on, growing into multicellular organisms. At maturity, then you can have meiosis happening to create the gametes that are haploid. And so everything down here would be diploid, or 2N. Up here would be haploid, or N. And again, this is a human biology course, so we could talk about 46 and 23. The process down here is mitosis. And the process to create those would be meiosis. So like I mentioned, there is a crash course on meiosis that you could watch both of them. It gives you a good overview. We're not going to go through the phases of meiosis in as much detail. Like I'd said, unlike mitosis, meiosis has two divisions. So it goes through meiosis 1 and meiosis 2. Meiosis 2 is the most like mitosis, because the chromosome number remains unchanged. The key thing about meiosis is we want to go from a diploid cell to a haploid cell. And specifically, that happens in meiosis 1. The chromosome number is reduced in half, and specifically in metaphase 1. And so we'll talk through briefly the processes of meiosis, but really hopefully focusing on how it's different and sets us up for our conversation about genetics. So meiosis 1, we have prophase. Much like prophase in any other part of cell division, we have chromosomes coiling and then they are visible. We have the homologous chromosomes coming together as pairs. That was something that was a little different than mitosis. In that in mitosis, they line up singly. So each pair has 4 chromatids, is called a tetrad. So these are somewhat interchangeable. Homologous chromosomes, homologous pairs, or a tetrad. Non-sister chromatids can exchange genetic information by crossing over. We're going to come back to that. In metaphase 1, this is different. So tetrads align at the equator. So you have 4 sister chromatids lined up, or 2 chromosomes. In anaphase, they're going to separate, and this whole chromosome is going to go to one cell, and this whole chromosome is going to go to the other. This is where the cells become haploid. You've reduced the number of chromosomes. Telophase, much like the process of telophase anywhere else. And this is illustrating that first bit. So key things that are different here is how they pair up. So this idea of tetrads and how they align in metaphase, and the fact that we're going from diploid to haploid coming up. And this is key about this first part of meiosis, or meiosis 1. Meiosis 2 is much more like mitosis. There's no more chromosome duplication. So that only happens once in both of these. And basically, I summarize them pretty quick. So prophase 2, chromosomes coil, and you can see them. They line up along the middle, and again, this time singly, like they did in metaphase, or sorry, in, oh, so many words, in mitosis. We have anaphase 2 where the sister chromatids separate. So this one moves over that cell and this one moves over to that cell. And then they ultimately make 4 haploid cells as an end product here. Again, here is an image that helps you kind of follow that flow through. So the idea that there's a lot of variation can be explained by a few key things. And so in this process of meiosis, how things line up. So in all of those images, again, not in your notes, but in the slides, things are color-coded as red for maternal chromosomes and blue for paternal chromosomes. And so it's absolutely luck of the draw in the formation of these gametes, in the formation of sperm and eggs, how they line up. So you might have, I'll switch colors on this one, you might have a maternal and a paternal. You might have a paternal and a maternal. And then another maternal on this side and a paternal. So even just with 3 chromosomes, they could line up differently, right? Because this is how they're going to separate to make the 2 new gametes. And so that's what we're talking about, independent orientation at metaphase 1. So it's the number of possible gametes formed. So each chromosome lines up independently and aligns at the equator. There's an equal probability of the maternal or the paternal chromosomes facing a given pole. The number of combinations for chromosomes packed in is 2n, where n is the haploid number of chromosomes. So in humans, we're talking about 2 to the 23. This is the number of possible different eggs or sperms that are created. I'll leave it for class or you to calculate this number. Now, that's not the end of creating a new offspring through meiosis and sexual reproduction. So it really depends. In this huge number, there's a random fertilization. Now, I'm not telling you to fertilize your egg cells or fertilize randomly. Hopefully there's some thought to it. But the idea that any 2 cells come together, so a random coming together of gametes, creates a huge amount of genetic variability. So I think statistically, and you can correct me if I'm wrong, this number then squared, I think, is then the number. So the likelihood of you creating 2 children that are identically the same from different pregnancies is impossible. It's just so improbable. Mathematically, it's not going to happen. I'm not talking about twins here. I'm talking about like 2 individual fertilization. So you have independent orientation and random fertilization explains so much of the genetic variability that we see in humans and in all sexually reproducing organisms. So this is illustrating it, the different combinations, this idea of independent orientation, how things line up. And you can see the different combinations here. Crossing over further increases genetic variability. So it's another cause of variability. Crossing over was listed up in our explanation of meiosis. So when we have homologous chromosomes or tetrads, they basically can get tangled up. And so I often do this. I stand, so this is a human, and I stand like this. And then I pick somebody from the group and they stand up like this, and we illustrate homologous chromosomes. They would be linked for their overall size, their centromere location, right? And so this idea that this is a double chromosome and this is a double chromosome. And so when they align as a tetrad, sometimes, if you will, their forearms get tangled up. And so if we were to tangle up and cross over, this part of my forearm would be from that homologous chromosome, and they would walk away with my forearm, if you will. This is so much better when I can act it out. I hope you're following along. And if not, the notes should have our back. So crossing over involves genetic exchange of material between homologous chromosomes. And so here we have this idea of them crossing over from that point up. And so you can exchange that material. So corresponding amounts of genetic material are exchanged. So it's not even a clear-cut number like that 2 to the 23rd power. That's not even accurate. It's a bigger number than we found because it's not always a clear orientation of maternal and paternal. You have this intermingling, this crossing over that creates further genetic variability. So if we were to kind of roll it all together, the things that are the same about mitosis and meiosis and the things that are different would be that they are both cell division. They both have chromosomes being duplicated once during the S phase of interphase. They do have a similar series of steps, but they're not the same. So you could list that they have a similar series of steps, but I didn't put it in the notes here. In mitosis, if we take a look at mitosis, the purpose of mitosis is for growth and repair in humans or reproduction if you're a single cell organism. There is one round of cell division, and what gets created are two daughter cells that are identical to the parents. It makes somatic cells, and remember those are body cells, that are diploid. And diploid in humans is 46 chromosomes. So meiosis is different. It's for different functions. It's used to make gametes or sex cells that are haploid. So this would be 23 chromosomes in humans. It creates 4 haploid daughter cells. And so each cell has half of the genetic material. They're not identical. And we have this idea of variability further being increased by crossing over. The other thing that are things that are different is that there's 2 rounds of cell division. So we have meiosis 1 and meiosis 2. And that other key bit, that stage that allows it to go from diploid to haploid, this metaphase 1 is how the chromosomes line up. So the chromosomes line up as tetrads here. They line up singly here. So in metaphase, chromosomes align singly. And again, I'm not sure if that's a word. Singly. Single file. That one's better. All right, so a few connections with this stuff, and then hopefully we'll transition into the genetics and you can understand that a little bit better because of this. So connection, there's so many things that can be wrong in this process of meiosis. So errors in meiosis can lead to genetic differences in us. Non-disjunction is the first one. So this idea that we have the chromosomes lining up as homologous pairs, but instead of separating like they're supposed to, they don't. We get 2 chromosomes being sent to the cells. So they both go that way, and there's nothing going that way. So the homologous pairs or sister chromatids don't separate properly. So it could be this one, or it could be in metaphase or anaphase 2, where they don't separate here. In this one, hopefully my diagrams are making sense. So in this one, you could have instead everything going this way and nothing going that way. So sister chromatids down here or homologous pairs, either one could have non-disjunction. So this could be anaphase 1, this could be anaphase 2. So what this results in is gametes with an abnormal number of chromosomes, and one of the examples of this is trisomy 21, which relates to a condition that you might recognize called Down syndrome. And so in this, it's non-disjunction because there's actually, instead of 2 chromosomes in the 21st spot in the karyotype, there's actually 3 chromosomes in that 21st position. So you remember the karyotype, right? And we had them from tallest to smallest all the way through, and we had 1, 2, down in the 21st spot, you would have not just 2 chromosomes, but you would have 3 chromosomes. And so you end up with an extra chromosome, or 46 chromosomes, in every cell in that individual. So non-disjunction is the failure of chromosomes to separate either in meiosis 1 or 2. It's not just chromosome 21. It could also be in not an autosomal chromosome. So these ones were autosomal. These ones could be in sex chromosomes, or the 23rd pair. And so sometimes you have non-disjunction. In this case, you'd have XXY, so an extra X chromosome, but that would be a male. You could have double Ys. There would be no syndrome. This would look like a normal person. No abnormality of note. I mean, genetically anyway. You could be non-disjunction and be 3 X's in the 23rd pair. And again, that would look like a normal female. And then you could also, as a female, be missing the second. And so this is a Turner syndrome, and is a recognizable genetic abnormality. So here's an illustration. So you could have non-disjunction of the tetrads, or non-disjunction when those lined up singly. And so here we can have what I was trying to draw, non-disjunction of the tetrad, so that's meiosis 1, or non-disjunction of the sister chromatids, and that would be meiosis 2. Non-disjunction is not the only thing, so other genetic abnormalities or differences or variation, honestly, it could be that, in this process of meiosis, you could have missing information, so deletion of genetic material. You could have duplication, so you have an extra or a repeat section of chromosome material. You can have inversion, so, a snippet of genetic material is put in backwards. And that's going to be different. You can have a translocation, so maybe this snippet gets moved over to here. And so all of this, there's so many things that can happen genetically that do result in differences or alterations of the chromosomal structure. Altered chromosomes can be carried by gametes, which can cause birth defects, or abnormalities can be in somatic cells, which could lead to cancer in the individual. So it could be, non-disjunction in mitosis or in meiosis. We seem to focus on it in meiosis because that's what's going to create new individuals, but it could technically happen in your cells, and it could lead to genetic abnormalities. All right, so chapter 9 will be about stuff that we can see again and in genetics. And hopefully what you take from this discussion on mitosis and meiosis is a real deep understanding of mitosis, but then an understanding of meiosis so that we can build on it for our conversation on genetics, and also that idea of how are these two processes the same and how are they different.